So good afternoon, everyone, and thank you so much for joining us today. I'm Esperanza Stanchoff with Humane Extension and Maine Sea Grant, a NECAN steering committee member and chair of the Outreach and Education Working Group for NECAN. I'd like to welcome you to the fourth webinar in the NECAN Sea Grant series. In 2016, the Northeast Sea Grant Consortium, in partnership with the NOAA Ocean Acidification Program, funded 4 research projects to address the impacts of ocean acidification on key resource species in the Northeast as an aid to assist coastal communities in adapting to current and future OA conditions in the region. The research and observational priorities defined in the publication of a NECAN paper in Oceanography magazine titled Ocean and Coastal Acidification off New England and Nova Scotia were used in the 2016 request for proposals. 4 projects were chosen and funded in Connecticut, Maine, and New York, and each of these webinars' focus is on one of those projects, their successes, challenges, and results. The full webinar schedule can be found on the NECAN website, and each are recorded there as well and posted there as well. Throughout the presentation, if you have any questions, you can submit them through the Questions tab on your GoToWebinar control panel, and we'll go through each of those during the Q&A session following the presentation. Today, we'll be hearing from Dr. Dianna Padilla from Stony Brook University on the response of blue mussels to ocean acidification. Dianna Padilla is a professor in the Department of Ecology and Evolution at Stony Brook SUNY University in New York and joint faculty member with the School of Marine and Atmospheric Sciences. She works on marine and freshwater invertebrates and algae, including ecology, conservation biology and restoration, functional ecology, phenotypic plasticity, invasion biology, and impacts of environmental change, including ocean acidification. She has 2 bachelor's degrees from the University of Washington in biological oceanography and zoology, a master's degree from Oregon State University in zoology, a PhD from the University of Alberta, Canada, and was a postdoc at Cornell University. Dr. Padilla held positions in the Department of Zoology, University of Wisconsin-Madison in 1989, and Stony Brook University in 1998. She's an American Association for the Advancement of Science Fellow and an Aldo Leopold Leadership Fellow in Conservation, a program developed to facilitate communication between scientists and non-scientists. She has also served as a program director in biology at the National Science Foundation. With that, I'm pleased to hand things over to Dianna, and she can take you through her presentation.
All right. Well, thank you very much, Esperanza, and thank you all for joining us today. So I'm going to be talking today about our project looking at blue mussels and changes in ocean chemistry. Oops. Make sure this goes forward. And first, I want to make sure to acknowledge my co-PIs / co-conspirators on this project, Lisa Milkey and Shannon Messick from the NOAA Fisheries Service. They were instrumental in helping design and carry out this entire project, and then a whole fleet of people who've assisted. This was a pretty giant undertaking that had lots of complex moving parts, and the folks at the NOAA Fisheries Service in Milford were just amazing in collaborating with everything. And then there were a whole lot of students, both graduate students and undergraduates at Stony Brook University, and a couple of high school students from Brentwood High School on Long Island that were all really important in producing this research. So we all know, we have to have our little introductory slide, that the amount of CO2 in the atmosphere has been increasing through time. That is reflected in the increase in CO2 in our oceans, which then drives a decrease in the pH of our global oceans. And that approximately 1/3 of all that anthropogenic CO2 in the oceans has been changing the carbonate chemistry of marine waters. We know that from a variety of research that we expect calcified animals in particular to have negative impacts when there is ocean acidification, particularly because of the carbonate chemistry changes and the saturation state of calcium carbonate decreases. We also know that shoreline communities are already experiencing wide ranges in pH and changes in carbonate chemistry, especially where we have areas of upwelling or there's lots of estuaries, such as the Atlantic coast. So coastal acidification can be more extreme than ocean acidification that we see out in the open oceans. Now, the negative effects that we expect of this changing chemistry in our oceans is changes in survival of organisms, their growth, aspects of their energetics or metabolism, their reproduction, their development, and behavior. So all of these different kinds of responses have been seen in organisms. And the questions that we wanted to address is there capacity within a species to adapt to this changing world? Are there differences among populations in their response to ocean acidification? Are animals that are currently facing stressful conditions better able to cope with OA stress? And when we see differences in robustness to ocean acidification, is it due to differences in acclimation by different populations? Is it due to phenotypic plasticity? Or are there genetic differences that drive differences that we might see? Or more likely combinations of the above. So we've been focusing on the blue mussel, Mytilus edulis. This is an aquaculture species further north, and there's interest in developing it as an aquaculture species in various parts of the Atlantic. It has a very, very wide geographic range and dispersal larvae. The larvae are generally in the water anywhere from 3 to 4 weeks. Now, it has been the focus of genetic and physiological studies for decades. So we actually know a lot about this species and how it responds to different environments. It's also readily grown and spawned in the lab and has a relatively short generation time. It can become reproductive in a year. Its life history, like most bivalves, their males and females free spawn their eggs and sperm into the water where there's fertilization. Embryos develop into feeding veliger larvae. The larvae start feeding at the D stage and then continue to grow and develop and ultimately become petaveligers, develop a foot, the velum shrinks, and ultimately they attach to the bottom and when they metamorphose. Now, what are the conditions like in the field where these animals grow? Now, we focused our work on populations around Long Island Sound. So populations at the mouth of Long Island Sound, which essentially experience oceanic type water, and then those further to the west. And what we found, just doing some weekly snapshots, sampling before sunrise, at noon, and just after sunset, just looking at what kinds of range of pH and aragonite saturation that we see at the different sites. And what we see is that summers have worse conditions or more ocean acidification, coastal acidification type signature than the fall. And we just had one sampling date in the winter, but the conditions get better as the winter progresses. But we can see that across sites, we see that all of the sites experience low pH at some time, and some of the sites, as we go west, have far lower aragonite saturation than others. So we know that pH and aragonite saturation vary seasonally. Summer's most stressful. It varies over the day, with the lowest generally being seen just before dawn and the highest pH just after sunset. The biggest single day change, though, we saw that at the temporal scale of 1 day, there can be very large changes in pH within 1 day at each of these sites, as well as changes in aragonite saturation over 1 day. So animals out there are presently experiencing low pH and low aragonite saturation at some sites, but not others. Now, there are different approaches to conducting ocean acidification experiments. One way is to add different constant amounts of carbon dioxide to establish your treatment levels. So the challenge with this kind of approach is that if you have a change in alkalinity of your source water, which most of us do, this will then change what your treatments actually are through time. So as the alkalinity changes day to day or over weeks, your treatments will be different from each other, but they will not be tied to particular carbonate chemistry. The alternative is to have fixed treatments to specific carbonate chemistry values, and this is what we did in all of our experiments. The challenge with this is that it's much harder to accomplish. You have to remove the CO2 from the water and then add prescribed amounts of CO2 to hit your target treatments. So it's a bit more of a challenge to create this, but then this allows us to really compare specific carbonate chemistry treatments within and among our experiments. Now, all the experiments we conducted were conducted under very similar conditions, anywhere from 18 to around 19 degrees C, with 3 different OA treatments. Now, we had a target OA treatments that were all, as I said, tied to the aragonite saturation state. So we had a high pH, which is similar to what you would expect in the open ocean, a pH of about 8 with an aragonite saturation of 2, a medium pH of about 7.6 with an aragonite saturation close to 1, and then a low pH, which projects basically future conditions as ocean acidification becomes worse, a pH of about 7.3 and aragonite saturation of 0.7. So what we did initially is we collected animals from around Long Island Sound along this expected gradient in stressful conditions, where in the west we have more eutrophication, more industrial pollution, annual dead zones, while in the east it's much less stressful and animals are more exposed to open ocean conditions. Now, our general experimental design for this study was an across-generational experiment with multiple populations. So we collected animals from each population from the field and then conditioned animals under 3 different OA treatments: our high pH, our medium pH, and our low pH treatments. We then spawned those animals and reared that first generation offspring under those same 3 treatments. And then we grew those animals up to produce a second generation of animals. I just realized that my pointer is wandering around, so I'll try and not touch it. The second generation of animals that then were conditioned under those same conditions, and then we grew up their larvae under each of the 3 different conditions to allow us to see if in the second generation do we have an improvement or the same performance as their first generation parents. So we separated mussels by population conditioning pH. We cycled them through warm and cold and filtered seawater. We played inspiring music. We did lots of things to try and get them to spawn. Once individuals spawned, we isolated them in a beaker of filtered water so that we could combine eggs and sperm, know how many parents we had, and isolate those individuals who did spawn for potential future molecular identification. So we combined the eggs and the sperm from all individuals within a treatment, within a population, and their test pH, the conditioned water for each test pH, and we allowed them to develop for 48 to 60 hours, depending on the experiment, before we then set up the experiment. So we used D-stage larvae, which is when they begin feeding. That's when we set up the experiment. The experiment was set up such that we had 300 larvae per liter in each replicate. Our replicates were beakers with screenings in bins with treatment water. So we had one population per bin, one beaker per population per bin. We had 5 independent replicates of every population and OA treatment. So the screening allows the water to exchange from the beakers but keeps the larvae within the beaker. So the bins with 1 replicate beaker from each population and conditioned water and carbon dioxide bubbled in to maintain the treatment level during the experiment were set up in temperature controlled bath so that with circulating water so that they would stay at our test temperature. And then treatments were interspersed across the tray so that no 2 replicates of the same treatment were adjacent. And this would account for any gradients that there might be in temperature or lighting or other things that might influence our results. Larvae were fed daily a mixture of Pavlova lutheri and Isochrysis galbana at a concentration across the bin of 4 × 10⁵ cells per mil per day. Bins were cleaned and refilled with new water every 2 to 3 days so that we could maintain our target chemistry values. All larvae are counted and individually pipetted into clean beakers on those days when the bins were cleaned and refilled. Water chemistry was tested before it was added to the bins and just before the water exchange. So after the animals had been in there for a maximum amount of time. And across the entire experiment, the variance in our chemistry was less than 2%. Now, the actual temperature that we measured during this first experiment averaged 19.68, and our measured pH and aragonite things, we came close to our targets, but we were a little bit lower. Our high pH was 9.7 with an aragonite saturation of 1.53. Medium pH 7.57 with an aragonite saturation of 0.77, and our low pH was pretty low. It was 7.26 was the pH and the aragonite saturation of 0.39. Now this is an example of our survivorship curves, and then I'll show you the rest of them in a second, but I just wanted to walk you through this first one first. We started out with 300 individuals in each pH treatment, and then we followed them through time and counted the number of individuals as the experiment progressed. And we had first metamorphosis in our pH 8 and 7.6 treatments at 21 days. The animals in the 7.3 treatment did not survive to metamorphosis. But you'll notice for Milford, we had higher survivorship at the medium pH than at the high pH. Now, if we look across the 4 populations that spawned, our 2 populations that we had further to the west never spawned, so we're not included in these experiments. What we can see is that, again, for Milford, we had highest survivorship in pH 7.6. For Groton and Orient, there was no difference in survivorship at 8 or 7.6. For Stony Brook, they were the total wimps. No one survived to metamorphosis in pH 7.6, but they did at 8, but we had much lower survivorship than the other populations. In all cases, we had no survivorship in the 7.3 treatment. Now, because we have very high temporal resolution of our sampling, we used survivorship analysis, or a form of accelerated failure time models, to test for differences in our those survivorship curves rather than initially just choosing an arbitrary date where we compare them. And what this analysis is very, very nice because it accounts for censored data. So we look at, how many days was it till an individual died, and we can account for those individuals that never died, that lived to metamorphosis. So we can compare the shapes of those survivorship curves. And we also did this for the time to metamorphosis. So these are those survivorship curves by pH for the 4 different populations that we looked at. And what we found is that there was a highly significant effect of pH. Everyone died in all populations in the lowest pH. There were differences at the other 2 pHs in survivorship. There was also a highly significant difference among populations. Populations were responding differently, and a significant difference in population by pH interaction. So different populations respond to pH differently. We can also, for the results of these tests, we get a hazard ratio, and the hazard ratio compares what's happening between 2 or more treatments. So the top 3 panels are for pH, and the reference, if we use 7.3 as the reference, we can see that we have much higher survivorship in 7.6, and in 8 we have the death rate is 0.28 times higher, is lower than we had much lower mortality in the 7.6 and the 8 treatment than in the pH 7.3, but across the entire experiment, there was no difference between 7.6 and 8. If we look across populations, we see that Stony Brook had the very lowest survivorship of all the populations, and the other 3 populations had relatively similar survivorship. Now, if we take that one slice in time, so you can get sort of a better picture of what the variance was like and what our results were, we can see that prior to first metamorphosis, so this is 19 days, this is the sampling date before anyone metamorphosed, what we see is that there was no difference between pH 8 and pH 7.6 in survivorship for either the Groton population or the Orient population. So for those 2 populations, there was no effect of pH. For the Milford population, animals only survived highest in the lower pH. It's pH 7.6, and poor Stony Brook, animals only survived in pH 8. Now, larvae began to metamorphose at 21 days post-fertilization, and we saw this in all populations and in all pH treatments for each population. So then we could use that temporal resolution of when individuals metamorphosed to then look at if there was a difference in development time. So these are cumulative metamorphosis curves for each population and each pH for 7.6 and 8, pH 8, because no one survived to metamorphosis in 7.3. And then to test whether these curves were different, the shapes of the curves were different, what we found is that there was no significant effect of pH. So pH did not influence development rate. All the populations, for all populations across pH, there was no effect of pH. There was a significant population effect. So there was a difference in populations in how rapidly they developed, but no pH by population effect. So these are our pH and population cumulative metamorphosis curves. And what we see is that there was, again, no difference between the 2 pHs, but animals from Groton basically developed the fastest, and those from Orient the slowest. And those from Milford and Stony Brook were in between. So there was a very big difference in how quickly animals developed, but it was not affected by pH treatment. So again, if we look at that cumulative number that metamorphosed, what we see is that for Groton and Orient, there was no difference between the two pHs in terms of response, in terms of who metamorphosed. In Milford, more in the lower pH, the 7.6, and in Stony Brook, again, we only had animals in the pH 8. So we grew those animals up for a year and then spawned them again to look at what happens in that second generation. Now, since we had no one in the low pH survive, this was only for the high and medium pH. And again, we spawned them as we did before. And this is what we found. These are those, again, those survivorship curves that you saw before. And again, in some cases, there is no difference between effective pH, but in some populations there was. For Milford, we had Milford animals that were grown at the high pH, and Milford animals were grown at the medium pH, and then tested across all three. And then for Stony Brook, we had those that were just in the high pH, and Orient, those that were just in the high pH. The other source populations, we weren't able to get to spawn in this experiment. We did conduct it again where we got everybody to spawn, but those data are still being analyzed. So what happened with the next generation? Well, as I said, not everybody spawned, but of those that did we see an improvement in performance based on the pH that they were grown at? And how were they affected by the other two pHs, or the other three? So if we start with Milford and we look at the Milford population, that the survivors from 7.6 in our first experiment, and we tested them at 7.3, 7.6, and 8 again. What we see is that, again, no one survived at pH 7.3. There was a slight but not significant improvement in performance at 7.6, but there was lower performance of those animals in pH 8. So they performed better in the pH that the parents were from. What if we look at the pH 8 guys? What we see is that there was a slight increase in performance in both pH 7.6 and pH 8 for those guys who were the survivors of the pH 8. So again, a slight increase, but marginally significant. For the Orient animals, we had Orient animals, the Orient 8 animals, and we saw the same or slightly reduced performance in their F2s in terms of survivorship. And the biggest response that we got was for our Stony Brook animals. Remember, the Stony Brook animals had very low survivorship and only in pH 8, and we had very much increased survivorship at both pH 7.6 and pH 8 in the second generation. So mixed results. They all didn't read the papers on what they were supposed to be doing, but they're sort of intriguing to think about. But again, we see differences among populations. So the results from our F2 experiment suggest that there are possible genetic effects as well as maternal effects. So with just genetic effects, we would expect increased performance in the pH in which they were reared in. Maternal effects, differences between the F1s and the F2s, and we see both of those. So what about differences in performance after the larval phase? So once all of our F1 juveniles were about 1 millimeter in size, which was at about 45 days after fertilization, then we were able to put them into small cages into a flow-through system so they would have access to natural plankton and we wouldn't have to change their cultures every 3 days. And so they were held under the same target OA conditions throughout that time. And so we had 3 flow-through bins for each treatment. As the animals grew, we moved them from small cages into larger cages, and always kept them at a density where they were not crowded. They were rotated among each of the bins every 1 to 2 weeks, so that once to 2 times a week, so that everybody was experiencing the different replicate bins for the pH. And then they were photographed 3 times during their first year so that we could assess growth. So over that first year in the flow-through OA system, we had we had the same target OA treatments, and we came close. We had a pH of 7.7 in our high treatment with average aragonite of 1.2, medium pH of 7.57 with an aragonite saturation of 0.79, and the low pH with a pH of 7.26 and an aragonite saturation much lower at 0.5. So what did we find in terms of survivorship? Well, this surprised me in that we had one individual of all of the animals that we reared that died within the first year, and that was it, just one animal. So essentially there was virtually no mortality across any of the pH treatments, across any of the populations in that first year. So then we wanted to look at growth rate. Now, growth rate is a very good integration of a range of physiological factors for animals. And for OA stress, what we would expect that there would be differences in growth rate due to limits on calcification at lower pHs. And so animals at lower pH should have slower growth rates resulting in smaller size. And I want to acknowledge Flor Villanueva, who was a high school student from Brentwood High School and is presently an undergraduate at Yale, who helped with these data. So initially we looked at shell shape, saying, was there any difference in how animals grew? And fortunately, there was no significant differences among populations between treatments or population by treatment interactions in shell shape. This made it much, much easier then to compare growth. Therefore, we just looked at growth in terms of the maximum shell dimension, shell length. So initially we looked at what was their growth rate for the first 3 months post-metamorphosis and asked, are there different, what do we find? Well, this was sort of interesting because we did find differences among populations, differences between the pH treatments, but not necessarily as one would predict, and we found population by pH differences. So for animals from Groton, the animals that were in the lower pH treatment grew faster than those in the higher pH treatment. For those from Milford, I'm sorry, I did that backwards. I did that right. For Milford, those in the pH 8 grew faster than those in the pH 7.6. For Orient, there was absolutely no difference between them. And for Stony Brook, we only had pH 8 animals, and they were sort of in between. They did fine. We looked at their size after 1 year. So this is basically where they're coming to asymptote at maximum growth. And this is when they're becoming reproductive. And what we found was pretty interesting. So we found that after 1 year of growth, there was no significant difference between animals grown at pH 8 or pH 7.6 for Groton or Orient. There's a slight trend at Groton, but it was not significant. For Milford, animals grown at 8 grew faster than those at pH 7.6. The Stony Brook animals grew the fastest. So what about other effects on their shells? So with increased ocean acidification, or that lower pH, we expect thinner and weaker shells in mussels, but we also expect faster growth to be associated with thinner shells. So I want to acknowledge Leonardo Campos, who again was a Brentwood High School student who helped with this work, and he's presently an undergraduate at Columbia. Now to do this, we had to cut the shells in half, embed them in resin, and then cut them lengthways along that maximum shell dimension to create a thin section that was then mounted on a glass slide and ground and polished so that we could create thin sections of the slide. We then took pictures of it through a microscope and stitched those pictures together so that we could use image analysis to quantify thickness and shell microstructure. So what did we find? Well, there was a significant difference among populations, but no significant difference between pH 8 and pH 7.6 on shell thickness. And there was no population by pH interaction. So shell thickness was pretty much just different for animals from different populations. Now, one of the curious things also was that the fastest growing mussels had the thickest shells. So these guys clearly didn't read those papers on shell growth either. So for Groton, Milford, and Orient, no difference, no effective pH. Thickest shells at Stony Brook and Milford, even though they were the fastest growing. So we found when we looked at shell microstructure, perhaps rather than affecting shell growth overall, there would be differences in the different allocation to different microstructure. Now, nacreous has more protein in it and therefore is generally considered to be more expensive shell material for bivalves to make. So we looked at what about the thickness of the nacreous layer, and we found no significant effect of population or ocean acidification in terms of the nacreous layer. So the nacreous layer was not different among animals from different populations or from different pH treatments. What about the prismatic shell layer? Now, the prismatic shell layer is generally considered cheaper to make. It has less protein. And here we did find a significant difference among populations, but no significant effect of pH or a population by pH. And again, the Stony Brook that were growing the fastest had the thickest prismatic layer. We also wanted to know if there was any effect on the strength of the shells. So therefore we used an Instron materials testing machine to determine maximum force required to break the shells. And what we found was that there was a significant difference among populations in shell strength, where Stony Brook was the strongest and Orient was the weakest. We found no significant difference as a function of pH. However, there's a slight trend maybe for the Orient and the Milford, but that's not significant. And we did look overall, was there a correlation between shell thickness and maximum load, how much force it took to break the shell? And there was a trend, but this trend again is influenced more across populations than within a population. And so this would require a much larger sample size, but again, it suggests that there really is this population signature rather than an effect of acidification. So to sort of summarize what we found for larvae across 2 generations, if we look at larval survivorship, there was a difference among populations. In response to lower pH, we found decreased survivorship, the same survivorship, or increased survivorship. But it was expected that we would have decreased survivorship. Oops. So that is not always as expected. For the time to development, we expected a decrease in time to development. We found no difference in time to development except for among populations. So again, a population level difference, but no effect of pH. In the 2nd generation, if we look at survivorship of the 2nd generation animals relative to the 1st generation animals in those same treatments, again, we found significant differences among populations. We found that sometimes they were the same or increased relative to their parents. If we were having selection, we would always expect increased performance in the 2nd generation. And for time to development, again, we would expect faster development in the 2nd generation, but what we found is no difference among pHs in development time. Again, we found a difference in between populations where animals from Orient develop at a faster rate. If we look at what we found for the juveniles, we found that there was no difference among populations in juvenile survivorship and no difference among the pHs in survivorship, even though we would expect lower pH to have a decrease in survivorship. For growth rate, again, we expected that lower pH would have an effect on slowing growth rate, but we only found that in one population. In most cases, it had no effect, and what we saw was, again, a difference among populations in growth rate, but not necessarily in response to pH. For shell thickness, we found differences among populations. It was not due to pH, even though we would expect lower pHs to create thinner shells. We would expect lower pHs to change allocation to different microstructures that might cost different amounts or be differentially soluble. The prismatic layer should dissolve at a faster rate under low pH, but we saw no difference. But we did see significant differences among populations. And similarly for shell strength, we saw differences among populations, but there was no effect of pH, even though we would expect reduced shell strength under low pH. So most studies use animals from a single source. They look at them over one generation or over one life stage, and then we tend to make generalizations to an entire species from that. Our results suggest that when we look at different populations, each of them is telling us a different story. And the stories they're telling us are not always consistent with our expectations of what ocean acidification should be doing with regards to negative impacts on these animals. So what does this suggest for future work? It means that we really need to start looking at more animals from different populations within each species. We need to start looking at responses across life history stages. And unfortunately, we also need to do more tests across generations, which is really a challenge for animals with much longer generation times. And with that, I'm happy to take your questions.
So that was great, Dianna. Thank you so much. So the only question that we have right now that I see is that although the study focused on blue mussels, are the findings easily transferred to other species with calcifying shells, that is oysters, question mark.
The only way we'll know that for sure is when we start looking at animals from different populations. Now, with the student, I've done a review of looking at all of the literature we've accumulated on larval experiments done under OA conditions, which people do the OA conditions a little bit differently, but looking at cross OA to where they've reared them from fertilization to metamorphosis. And what's interesting is that we find a scatter of results. We don't always find consistent results. Now, sometimes that's because of when people started their experiment. One of the things that I didn't talk about that we did in another experiment is that if we exposed animals to higher than a higher pH, a 7.6 or an 8 pH during those first 2 to 3 days, and then grew them at pH 7.3, we did not see the same mortality. Instead, we had animals complete to metamorphosis. That seems to be something that is emerging across species, is that perhaps for bivalves, those first 3 days are the most vulnerable. But I think that a lot of the variants that we're getting in some species in terms of cross studies could be because different populations are responding differently.
Great. So the next question is, I missed some of the beginning slides, so pardon me if you stated this, but do you know the pH conditions at each population site? Are there any monitoring sites that give you the dial variation of seasonal variation in carbonate conditions?
So we have, let me just back up a little bit. Let me see if I can get back to the beginning. So we have a small amount of monitoring data, and then I know that Shannon Messick has more monitoring data for various parts of Long Island Sound. So this is just based on some sampling that we have. Definitely at Orient and Groton, conditions are more favorable. They see higher pHs and higher aragonite saturation than we see at Stony Brook or at Milford. We definitely need more monitoring and more continuous monitoring so we can get a better picture of what's going on. Conditions are definitely worse in the summer when these animals would be spawning. It gets better in the fall, and in the winter we just have 1 day of sampling, so we can't really characterize that.
So the next question is, can you re-explain the differences you see between maternal effects and genetic effects in regards to your work here? I'm not necessarily seeing the difference.
Okay, so let me get to okay, so if we had the biggest is what we see from Stony Brook. So if it was just genetic effects, then we would not expect to see that really big increase in survivorship in 7.6 from our pH 8 guys. Our pH 8 guys should have been the only guys that survived in the second one. So it suggests that, you know, our poor 7.6 guys that all died at Stony Brook, and we had this really, really low performance overall, even in pH 8 of our Stony Brook guys, is stripped away in our second generation. So the second generation guys really just about outperformed just about everybody else in many ways, but we would never have predicted that just based on the F1 performance. If it was just a genetic thing, then we still should have expected to see no survivorship in the 7.6, but that's not the case. We actually had very high survivorship in the 7.6 treatment, as well as a 7.8 and a really, really high one. For the orient ones, if this was pretty much genetic, then we would have predicted an increase in performance in the pH 8 when it was tested again in pH 8, and we did not see that. So it all, so it's all sort of, the Milford, we did see a slight increase in 7.6, and again that they decreased in the 8 from the 7.6. So that suggests more possibility of genetic selection, but the results from the Stony Brook population definitely suggest that there's a maternal effect, or at least I think it does.
Okay, do you have a sense of what could happen with pH conditions that change on a tidal daily basis?
So right, so this is one of the questions is that we all have, right? Is what are these animals really experiencing in the real world? And from the sampling that we did here when we looked at single day changes, as well as some other sampling that I've been doing subsequent to this, what I've seen is that the biggest tidal changes and single day changes seem to be more in pH than in aragonite saturation, and seem to be driven a lot by oxygen conditions. So I think that, again, this is going to be very site specific, and we need more monitoring and better temporal monitoring to really understand what the variability is that these animals are seeing in terms of both pH and especially the carbonate chemistry, because I mean we have pretty good convincing evidence, I think, from oysters that it is the carbonate chemistry that's most important for larval bivalves. And if that's the case, then that's really what we need to look at in terms of short-term temporal variability, in terms of a tidal cycle, as well as then across weeks, and especially during the time for whatever organism you're looking at would be when they're spawning. Does that answer the question, or does that just skirt it too much?
I think that's really interesting. We'll see if anybody responds, but I was wondering what's the future for this team, or are there any plans to continue this research?
Well, the future is, of course, to try and get more money, but but one of the things that we would like to start looking at is, okay, what happens when you have multiple stressors?
Mm-hmm.
So what happens when you're experiencing both OA stress and temperature stress or oxygen stress? How do those kinds of stressors, are they synergistic? Are they additive? What do they do? And I've started doing a little bit of work like that with the blue mussels on looking at temperature stress and food stress, and seeing that in some cases you can get, and putting it in a variable time, that variability itself can help ameliorate some stressors. But yeah, so that's what we'd like to do. We'd also like to get money to start looking at, let's look at the genetic background of these animals. We've saved samples of all of our parents, our F1s, our F2s, and so try and get the money to see, can we detect any genetic signatures? But both those lines of research require additional funding.
Right. Okay. Thank you. There's another question. Do you feel a diel variation in pH would mimic the overall low pH continuous effect? In other words, should we be accounting for diel pH variations to make sure our oyster hatchery has a higher survivability?
If I was a betting person, this is not based on data, right? This is just sort of a gut feeling looking at the data that we have, our observations in the field, is that unless oxygen is getting really low, that what's going to be most important is going to be the carbonate chemistry. And so just tracking pH can sometimes be misleading because it will be so affected by oxygen, where the carbonate chemistry not. So I think if one can measure the carbonate chemistry and see if you see a similar extreme diel cycle, and again, this is all going to be very site dependent. I mean, that's one of the things that is so interesting. Milford and Long Island Sound are just across the water from each other, right? We can see each other when we're standing on the shore, and but the conditions can be extremely different. And so we're talking about just, tens of kilometers making a big difference in what water conditions are like.
Great. So there's another question that came in. Is there the potential to also monitor the antibodies / immune system strength of the animals with changes in pH and across generations?
Well, that's the stuff that Bossum does. So I was going to say it could, but it not possible in the experiments as we were doing them. So what we wanted was we wanted to be able to have a lot of replication and be able to get good temporal resolution and replication in terms of, what we were looking at, and numbers of that requires that you limit the number of individuals per replicate in order to get enough animals to do that kind of study. You need a whole lot more animals, which makes it technically much more challenging to even attempt anything like we did. But I think that our results could indicate which populations and which pHs one might do a follow-up study on to focus in on those questions. Basically say, okay, how important are those physiological responses, and are those physiological responses in terms of the immune system, what are important potential drivers for the responses that we saw in our results. Mm-hmm. What was interesting is that what I was totally surprised by is the results that we got on shell thickness and shell structure. And the immune system does play an important role in building shells and maintaining shells in bivalves. But it was interesting that we did not see any indication that the net result of that, the shell structure and shell thickness, were being affected in mussels. That does not mean that that would be true across oysters or other species. I know that other work that's been going on juvenile oysters has found that acidification in the same range that we were looking at actually does impact shell thickness and shell deposition.
Great. So we do have one last question. I realize it's 2 o'clock, so if people want to hang on for another few minutes, that's great. I know Diana's willing to do that and to answer any questions further. But I just want to remind folks that that these, in case you do have to get off, that all of these webinars are recorded and available on NECAN.org under the resources tab. So I'll give you your last question, and then we'll finish up. Do the natural populations of mussels in those sites seem to be generally stable, or are they declining as rapidly as the ones on the south shore of Massachusetts?
So we don't have any monitoring data on populations of mussels in these areas, and so I really can't say exactly. You know, I talk to old timers and they always tell me, oh, we used to have a lot more mussels than we have now. I don't know that we could say that. What is true is that as you go further to the west, there's relatively fewer mussels than there are as you go further east, but there's also a change in substrate. So yeah, I don't know. It would be good to have more, not only the water quality monitoring, but the monitoring of the populations on the shore, so we could actually be able to track some of that. But to my knowledge, we don't have good monitoring of that.
Okay. Oh, we have one more. Actually, let's see. No, that was the last question. So that was the last question. So that concludes our webinar for today. And again, as this was the last webinar in our series, the recording for Diana's presentation and the other webinars in the series will be, as shown on this slide, will be available on NECAN's website. And I just want to thank you so much, Diana, for such a great presentation. And thanks to everyone, again, for joining us today. And that concludes our webinar.
Thank you.